Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome to the deep dive. Imagine being able to tell the health, state, and even what a tiny single cell is about to do, just by understanding how crowded its insides are.
Yeah, it sounds almost like science fiction, doesn't it?
It really does, like reading a hidden report card in something as fundamental as density. Even at this incredibly small scale, these physical properties hold, well, fascinating secrets.
And unlocking those secrets is exactly what drives a groundbreaking new development from researchers at MIT. They've developed a significantly faster, more streamlined way to measure the density of thousands upon thousands of individual cells.
Okay, so that's what we're diving into today. Using the source material you shared, there's a news article, and also the scientific paper describing the work itself.
Right.
Our mission, then, is to unpack this innovative new technique, explore the surprising biological insights it revealed,
There were definitely some surprises.
and understand why measuring density quickly and precisely could become, well, a powerful new tool for understanding biology and potentially even for predicting how diseases like cancer might respond to treatment.
A really big potential application there.
Let's get into it. At first glance, cell density might sound like a simple concept.
Mass over volume, right? Why is it so important?
Well, it's anything but simple when you look closely. Cell density is essentially the ratio of a cell's mass to its volume, yes. But what that number really tells you is about the molecular crowding inside. You know, how packed it is with everything from proteins and lipids to nucleic acids, all the cell's machinery.
And as a cell changes what it's doing, growing, maybe specializing, or perhaps encountering a problem, that internal crowding changes. Is that the idea?
Exactly. As cells change state, whether they're rapidly growing and dividing, differentiating into a specialized type, or even undergoing programmed cell death, they gain or lose water and other key molecules. This changes their internal composition and directly impacts their density. So tracking these often subtle changes can offer incredible insights into their function and their state.
But measuring these subtle density shifts, especially for, vast numbers of individual cells, has been a major hurdle in research.
It certainly has. The scale and the resolution needed presented a real technical challenge with previous methods. It's worth noting the lab behind this new work, led by Professor Scott Manalis at MIT, actually has a history in this area.
Right, they weren't starting from scratch.
No, not at all. They developed an earlier technology back in 2007 called the suspended microchannel resonator. It's quite a clever device.
Okay.
It could measure cell mass very precisely by detecting tiny changes in its vibration frequency as a single cell passed through a vibrating channel.
So that first device was great for mass.
Mm-hmm.
How did they adapt it to get density back then? Because density needs volume too.
Right. In 2011, they adapted the suspended microchannel resonator to measure density. But to do this, they had to pass cells through the device twice, sequentially, each time using a different fluid.
Twice. That sounds cumbersome.
It was. By measuring the cell's buoyant mass in two different fluid environments, they could calculate its absolute mass, volume, and finally, density. But as you can imagine, loading cells, running them through, then switching fluids and running them through again. It's quite slow.
How slow are we talking?
With that two-pass approach, they could only measure maybe a few hundred cells per hour.
A few hundred. Okay, that's not really high throughput.
Not at all for the kind of questions people want to ask now, which brings us to the breakthrough in this new study, how they dramatically increased that speed and made it more streamlined.
So what's the new setup?
They engineered what they call a fluorescence exclusion, coupled suspended microchannel resonator.
That's a mouthful.
It is. Let's just call it the new technique for now. What they did was combine their core suspended microchannel resonator technology with a fluorescence microscope setup before the resonator itself.
Okay, walk us through exactly how this combination works in one pass. That's the key, right?
One pass. That's the key. So cells flow through a microchannel that contains a fluorescent dye. Now, this dye is chosen specifically because the cells cannot absorb it. They exclude it.
Okay.
Just before the cell reaches the resonator part that measures mass, a fluorescence microscope images the channel. As the cell passes this point, because it excludes the dye, it creates a sort of shadow, or more accurately, a dip in the fluorescent signal.
Like a little dark spot moving through the break die.
Exactly. And the size of that dip is directly proportional to the cell's volume.
So you're using light and a dye to figure out the cell's volume first.
Precisely. You get the cell's volume optically as it flows through the fluorescent region. Then that same cell continues its journey into the suspended microchannel resonator part,
Whereas mass gets measured.
where its mass gets measured, just like the earlier device. Because they get both the volume and the mass measurement sequentially on that single pass through the device, they can calculate the density right away, much faster.
Got it. And the lead author, Richard Wu, emphasized this streamlined approach.
Yes, he highlighted that the goal was indeed this streamlined measurement, where cells only need to pass through the entire system once. Makes a huge difference.
And the numbers on the speed and precision are where this really shines, I gather.
They truly are. This new technique can measure the density of up to 30,000 single cells in just 1 hour.
30,000 compared to a few hundred.
Exactly. That represents a 10 to 100-fold increase in throughput compared to their previous methods. It's a massive leap.
And the precision is still high?
Exceptionally high. The reported precision for their density measurement is around 0.003%, which translates to 0.00003 grams per milliliter. Very, very sensitive.
Wow. Being able to measure thousands of cells so quickly and precisely must unlock entirely new ways to look at basic cell biology.
It absolutely does. And this is where we start getting into the fascinating biological discoveries made possible by this speed. One area they investigated was what happens to the density of immune cells, specifically T cells, when they are activated.
Okay, T cells, they're crucial, going from quiet to active.
Right. T cells normally sit in a quiet resting state, but they switch to a high-growth state when they're stimulated, like when they need to fight infections or diseases.
And what do they see happening to density during that critical switch using this new technique?
This was one of the really intriguing findings. The researchers discovered that as activated T cells transition, they gain both new molecules, building blocks for growth, and water. Lots of water, apparently.
Okay, so they're getting bigger, gaining mass.
But here's the twist. From their pre-activation state, day 0, to just 1 day after being stimulated, the average density of T cells actually dropped. It went from about 1.08 grams per milliliter down to 0.06 grams per milliliter.
They became less dense. Even though they were growing and gaining new material, that seems counterintuitive.
It does, initially. But the interpretation, as the researchers explain it, is that as the cells enter that rapid growth phase, they actually gain water faster than they gain other heavier molecules like proteins.
Ah, so the water dilutes the internal contents, relatively speaking.
Exactly. It essentially makes the inside of the cell less crowded in terms of mass per unit volume, even as the total mass is going up. Richard Wu is quoted as saying, "This is suggesting that cell density is very likely reflecting an increase in cellular water content as the cells transit from a quiescent, non-proliferative state to a high-growth state."
So cell density appears to change significantly during this activation process, and it might even be a signal, maybe a biomarker, related to how well those T cells are preparing to proliferate and do their job.
That's the potential implication that density acts as an interesting biomarker tracking this fundamental cellular transition. And it wasn't just T cells. They observed similar density drops during activation in other cell types they studied, including activated human naive B cells. And they even saw density changes in a specific mouse cell line when a crucial growth factor was removed, forcing a change in its growth state. So it seems like a potentially general phenomenon.
Interesting. Beyond tracking changes during activation, what else did they learn about cell density just by being able to measure so many cells so quickly? Did they look at, the normal variation?
They did. They took a close look at the variation in density within a population of cells that are actively proliferating or growing. You might expect something as fundamental as density to be tightly controlled by the cell, right?
I think so, yeah.
And they did find that in populations of dividing cells, density is indeed remarkably tightly regulated. The variation measured by something called the coefficient of variation.
Which is basically a way to say how spread out the individual measurements are around the average.
That's a great way to put it, yes. And that coefficient of variation for density was below 0.6%.
Which sounds pretty small.
It is, and importantly, this low variation is much smaller than the variations they saw in just cell mass or volume within the same population. Those varied a lot more,
Okay.
and it's also significantly higher than the precision of their measurement technique, which confirms that this tight regulation of density is a real active biological process, not just noise from the instrument. The cells are actively controlling it.
Okay, now tell us about the surprising finding regarding the distribution of these densities. I heard that didn't quite follow standard expectations.
Yes, this was quite unexpected and points to maybe a deeper complexity in how cells manage their density. When they plotted the distribution of single-cell densities across a population of proliferating cells, it didn't fit the standard statistical patterns you often see in biology, like a normal or Gaussian distribution, that typical bell curve.
The Mel curve, what did it look like?
Instead, the distribution was asymmetric and had what statisticians call heavy tails.
Heavy tails. What does that suggest about the cell's internal control system? Like more outliers than expected.
Exactly. It means there are more cells with densities significantly higher or lower than the average than you would expect in a standard distribution. This suggests that the mechanism controlling cell density might be more intricate than a simple negative feedback loop that just keeps bringing the cell back to an average density.
Because that simple control would usually give you the bell curve.
Often, yes. A simple thermostat-like control tends towards symmetry. The heavy tails indicate a more dynamic or maybe complex control system is at play. We don't fully understand it yet, but it's a clue.
And how did quiescent or non-growing cells compare in terms of density variation?
Interestingly, cells that were in a quiet, non-proliferating state showed a higher average density and also higher density variability compared to their actively growing counterparts.
So quieter cells are denser on average, but also more varied in their density.
That's what they found, which further reinforces the idea that the regulation of density is somehow fundamentally linked to whether a cell is actively growing and cycling through the cell division process. The rules seem different.
Okay, this is fascinating fundamental biology. Let's shift gears a bit to the potential applied uses. This kind of rapid, precise measurement capability seems incredibly promising for areas like, say, predicting how cancer cells might respond to drugs.
Absolutely. That's a major application area the researchers explored, and it's a really exciting one. A key goal in modern cancer treatment is what's known as functional precision medicine.
Right, testing a patient's own cells against drugs.
Exactly. The idea is to take a patient's actual tumor cells and test them outside the body to see which drugs are most effective for that specific person's cancer, rather than relying solely on generic guidelines or genetic mutations.
That makes so much sense, tailoring the treatment directly. What's the challenge there currently? Why isn't that standard practice everywhere?
Well, a significant challenge is that primary cancer cells taken directly from patients often don't grow or proliferate very well when cultured in the lab. They can be quite finicky. And if they do grow, it can take several days or even weeks to get enough cells and run standard tests that measure long-term growth or viability. During that time, there's a risk that the cells might change, becoming less representative of the tumor still in the patient.
So time is a problem, and the cells might not even cooperate.
Exactly. There's a real need for faster ways to assess drug response, especially ways that don't rely on the cells actively dividing in the lab over long periods.
And this is where rapid density measurement could come in. The hypothesis being that density changes fast if a drug is working.
Precisely. They hypothesized that rapid changes in cell density after drug exposure, maybe within hours or a day, could serve as that fast biomarker. They tested this concept using cells from a model of pancreatic cancer derived from a patient sample.
A specific, well-studied model.
Yes. This particular model, known as NIBRX1362, is well characterized. It's known to have a specific genetic alteration, a KRASG12D mutation, which is common in some cancers. And importantly, its response to certain drugs in living systems in vivo is already understood.
So they had a baseline to compare against. Which drugs did they use to test their hypothesis?
They treated cells from this model with two different cancer drugs. One called trametinib, which this cancer model is known to be sensitive to in a living system, and another called gemcitabine, which the model is known to be less sensitive to in vivo.
So one effective drug, one less effective drug for this model.
Right. And then they used their new technique to measure the single cell mass and density at very early time points, just 24 and 36 hours after adding the drugs.
Just a day or a day and a half later. So a hyperacute timescale. much faster than waiting days for the cells to grow or die in response to treatment.
Exactly. They wanted to see if density could give an early warning signal, and the results were quite compelling.
What did they find?
The changes they measured in single-cell density after drug treatment accurately reflected the known longer-term responses of these cells to the drugs seen in living systems.
So the density shift matched the known effectiveness.
Yes. Trametinib, the drug the model was sensitive to, caused a significantly greater density change compared to gemcitabine, the less effective drug, even at these early 24 and 36-hour time points.
Wow. So the density change was like a very early indicator of whether the drug was having the intended biological effect.
That's the key finding and the potential power of this approach. As White Richard Wu put it, cell density is a rapid biomarker to predict in vivo drug response in a very timely manner.
Did using density add predictive power beyond just measuring cell mass alone, which their previous technology could do, or was mass just as good?
No, density provided unique information. The analysis showed that combining both density and mass measurements provided a much more accurate prediction of the known long-term response than using either measurement alone.
Okay, so the combination is best.
The combination was best, but interestingly, density changes by themselves were found to be a stronger predictor of the known long-term response than mass changes alone in their analysis. Density seemed to capture something critical about the drug's impact very early on.
That really highlights the unique information density provides. It suggests this technique could potentially give doctors and researchers crucial information much faster for precision medicine efforts.
Right.
Even for patient samples where the cells might not be actively growing well in the lab.
That's the hope and the potential impact. Professor Manalis actually co-founded a company called Travera that is already working on using these kinds of mass measurements clinically, and the paper mentions they are incorporating density measurements into their efforts now, too.
So it's moving towards clinical application.
It seems to be.
Yeah.
And the researchers also mentioned ongoing work using this technique to evaluate other types of cells, like those used in biomanufacturing to synthesize therapeutic proteins, like antibodies. So its applicability might be quite broad beyond just cancer drug prediction.
Now, while the potential is clearly immense, it's important we also touch on the limitations. The researchers themselves are careful to point these out in the source material regarding this initial drug response study.
Absolutely, and that's crucial for good science. They're very clear that their drug response experiment focused on only one patient-derived cancer model, that specific pancreatic cancer model, NIBRX 1362.
Just one model. So future studies are absolutely essential to see if this density-based prediction holds true across the incredible diversity of different cancer types.
Exactly.
And the variations you see from patient to patient are cancer isn't just one thing.
Precisely. Cancer is notoriously heterogeneous, meaning it varies a lot. Responses can vary widely even within the same cancer type. So testing this approach against a much broader panel of cancer types and patient samples is the necessary next step.
What other limitations do they note?
Another key one they mention is that this study specifically looked at the density response to just two different drugs, trametinib and gemcitabine.
Okay, so just two mechanisms of action, essentially.
Right, meaning they need to do more research to see if changes in density can reliably predict outcomes for a wider range of cancer drugs that work through different biological mechanisms. Will density shifts always correlate with effectiveness regardless of how the drug works? That's still an open question.
But the good news is that the incredibly high throughput of this new technique makes those broader, necessary follow-up studies much more feasible, right? They could test many more models and drugs much faster now.
That's a huge advantage. What would have taken ages before can now potentially be done much more quickly, accelerating the process of validating this approach.
Okay, so summing up what we've explored today, we have this innovative, high-throughput fluorescence exclusion coupled suspended microchannel resonator technique. Quite a name.
Ah, yes.
That allows for the rapid and precise measurement of density for thousands upon thousands of individual cells per hour.
And this newfound capability has already revealed surprising insights into fundamental cell biology. It showed how cells tightly regulate their crowding or density as they transition between quiet and active states.
Right, with proliferating cells maintaining a lower, more tightly controlled density.
And also showing that density distributions in cell populations don't always follow the simple patterns we might expect, hinting at complex control mechanisms.
And importantly, on the application side, it demonstrated the really exciting potential of using these rapid density changes as a predictive biomarker.
A way to assess how patient tumor cells might respond to specific drug treatments, potentially much faster than current methods, even if the cells aren't actively growing in the lab.
Being able to measure this fundamental physical property of cells, their density so efficiently, truly opens up new avenues for basic biological research and potentially faster, more effective ways to guide treatments for complex diseases like cancer.
It really feels like adding another dimension to how we can understand and interact with cells.
Definitely. So what stands out to you after this deep dive? Maybe a final thought for our listeners to chew on. Consider this. How might this seemingly simple property of cells, their density, be a window into countless other cellular processes or disease states that we haven't even considered yet?
That's a great question.
Could routine density profiling become a standard tool in research labs and clinics, revealing secrets about cell health and function that are currently hidden? And what other subtle physical properties of cells are just waiting to be explored and leveraged with new technologies like this one?
It makes you wonder what else we're missing just because we haven't had the tools to look for it properly. Density might just be the tip of the iceberg.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.